to my YouTube channel Share, and welcome to the part 2 of seed structure and germination. And in part 1 we have discussed external and internal structure of a bean seed and many other basics of this lesson. So make sure you check it out, which I've linked in the description box below and also at the top of your screen. So now in this video we are going to discuss basics of a grain, internal structure of a maize grain, germination, condition necessary for germination, and types of germination. So let's get started. So guys, here first one, basics of a grain. So a grain is a fruit, okay, in which a fruit wall and the seed coat are fused together. Okay, for example, maize and wheat. Most of these grains will be monocot. Now let's see the internal structure of a maize grain. So guys, this is the internal structure of a maize grain. So here you can see this outer protective structure is pericarp plus seed coat, which means fruit wall plus seed coat. So as I told you, grain itself means fruit wall and seed coat are fused together. So this is the layer, fruit wall and seed coat are fused together. Okay, and this line layer here you can see. So this layer is called aileron layer, which is rich in protein. Okay, and this blank space here is called as endosperm, which we have discussed in our first video more detailedly. Okay, so the next one is this. As this maize grain is a monocot, so it has a special name to call, that is called a scutellum. So this maize grain is only of one cotyledon, so it is called as scutellum. Okay, so this has a plumule here and the radicle here. So plumule is protective by a protective layer called as coleoptile, and radicle is protected by a protective layer called as coleorhiza here. Okay, so plumule and radicle both have protective layers, coleoptile and coleorhiza. And this is the diagram of a longitudinal sectional of a maize grain. Let's go with the next diagram, which is this. Okay, so guys, in this we are going to discuss about the germination. So let's see the germination. So germination is the process of growth of a seedling. Okay, this is the process of growth of a seedling from an embryo. Okay, from an embryo till its end extent, that may be till its death. Maybe from a seedling to a tree, maybe from a seedling to a plant till its death. The process of its growth, it's completely called as germination. Okay guys, so the seed germinates only in its favorable conditions. Remember, only in its favorable conditions only the seed will be germinating. Okay, if there are unfavorable conditions, then the seed will rest inside the soil. Okay, then the seed will rest inside the soil, and this period of resting the seed, okay, this period, not the seed itself called, but the period, the complete period of resting the seed inside the soil is called as dormancy period. Okay, inside the soil in its unfavorable conditions, it is completely called as, here you can see, it is completely called as dormancy period. Okay, and the seed in this period, till now we have discussed the period. Okay, the complete period in which the seed will rest is dormancy period, and the seed itself is called as a dormant seed. Okay, and the seed itself is called as a dormant seed. Okay, next is conditions required for germination. As I told you, there are some particular conditions, favorable conditions, so that the seed can easily germinate. So here we are going to discuss those conditions. The first condition is temperature. So in temperature, we need optimum temperature for the germination of a seed. So the optimum temperature is between 25 to 35 degrees Celsius. Okay, so this is the optimum temperature for the germination of a seed. The next, water. So water is one of the very important things. So now let's discuss the process, how this water will help to grow out our plant. First, water will enter into the seed. First, water will enter into the seed. Then, this water will enter into the seed by the help of micropyle. Okay, we discussed about this micropyle in our first video, which I've linked in the description box below. Okay, so in micropyle, with the help of micropyle, this water enters, and because of the entry of the water, the seed swells. Okay, remember, because of entry of water, the seed swells. Okay, because of the seed swelling, the seed coat will get ruptured. Okay, the seed coat will get ruptured and allows the delicate plumule and radicle to come out and grow. Okay, so which helps to grow out the plant. Okay, so this much process is happening just because of the entry of water inside our seed. Okay, so the third condition is oxygen. Oxygen is very useful for respiration of a seed. Okay, as we also need oxygen, in the same way, oxygen is also needed for a seed to germinate and also energy for growth. Okay, so I want to tell you guys a thing. If you're going to keep a seed deep inside the soil, then the seed fails to germinate. Why? Because this process has three particular reasons. The first one, there will be no enough or correct temperature going in. Number two, there will be no enough oxygen. And finally, number three, the plumule and radicle are very delicate, so they can't fight with all these layers of soil and come out. So in the between of this process only, they will break and the seed will die. Okay, so the next is this three types of germination. Okay guys, so there are three types of germination here, as you guys can see. So first germination is hypogeel germination. So hypogeel germination, in which the seed and root, both of them will remain underground and only allows the plant to go in. Okay, this is called as hypogeel germination. Okay, so the next is epigeel germination. Epigeel germination, in which only the roots will be underground, including the seed. It will go above the soil. It won't remain underground. It will go above the soil like this, as shown in the picture. The third one is vivipary germination. So vivipary germination, which means that the seed will germinate when it is already attached to the plant or the fruit. Okay, so you can see this case in the tomato in the picture. So the seeds are inside the tomato only. We haven't kept them in the soil. We haven't kept them in water. But they already germinated inside the fruit itself. So this type of germination is called as vivipary germination, which will germinate inside the plant, inside the parent plant or the fruit itself. Okay, so the next. So answer all these tough questions which are hidden inside this explanation only. If you have listened the explanation correctly, you can answer all these questions. Answer them and comment me down in the comment section below. The people who comment right are going to get a heart as well as the shout out in my next video. So the first question, define a grain. Second question, which layer is rich in protein? Third question, which layers protect plumule and radicle? Fourth question, what is the dormancy period? Fifth question, what is vivipary germination? Sixth question, why is water required for germination? That's it guys, that's it about this video. Meet you in my next video, that is next coming Sunday. So hope you guys like it. So in my next video I'm going to come with one more interesting lesson and one more interesting subject. Thank you for watching. Bye bye.